Okay, so think about this. Your brain sends out these incredibly fast electrical signals, like little lightning bolts, telling your body what to do. But how do they actually get from a nerve cell to, say, a muscle cell? How do they jump that tiny gap between them? Because it's not like electricity just leaping across, right? If it tried that, the signal would fizzle out, lose like 99.99% of its strength. So clearly, evolution needed a much smarter solution. And that's exactly what we're diving into today. We're exploring the amazing world of cell-to-cell communication, focusing on a key chapter in Boron and Boulpaep's Medical Physiology, the one covering synaptic transmission and the neuromuscular junction. Our goal here is pretty straightforward. Break down how nerves talk to muscles and really show why getting this is so fundamental for anyone studying physiology, especially for understanding what goes wrong in disease and how treatments work.
Absolutely. It's like learning the body's internal messaging system. We'll start with the bigger picture. Why do we even need these specialized junctions, these synapses? Then we'll zoom right into the nitty-gritty molecular details. And importantly, we'll keep connecting it back to the so what. Why does this matter for clinical practice, for understanding patient conditions?
Right. So the big problem, that gap.
Yeah.
How did biology solve the signal jumping issue?
Well, that was a huge debate for a long time, back in the 19th century especially. Were cells physically connected, like little bridges between them, or was it all chemical signals floating across? It really wasn't settled until maybe the 1960s, with electron microscopes letting us see these tiny structures and neurochemistry identifying the molecules involved. And the answer was, well, both. The body uses two main strategies, electrical synapses and chemical synapses.
Okay, let's tackle the first type then, electrical synapses. These are the direct connections, right? The
Yeah.
high-speed ones.
Exactly. Think of them as direct wires. The membranes of the two cells get incredibly close, just a few nanometers apart. And they're studded with these special protein channels called gap junctions. Each gap junction is made of proteins called connexins, and they line up perfectly across the gap, forming a direct pipeline, an aqueous channel, connecting the inside of one cell to the inside of the next.
So the electrical signal can just flow straight through.
Pretty much. It's a very low resistance path. The voltage change just spreads directly, with almost no delay and very little signal loss, super fast. Most of these are reciprocal, meaning the current flows equally well in both directions. But some are rectifying, acting like a one-way valve, only letting the signal pass easily in one direction. You see these where speed and synchronization are absolutely critical, like in heart muscle or certain reflex pathways.
Okay, direct, fast connections. But you said there's another way, the chemical way, and this sounds like where things get a bit more complex.
They do, but also much more flexible, which is essential for the nervous system. Chemical synapses have a wider gap, maybe 30 to 50 nanometers. And the key feature is on the sending side, the presynaptic terminal. It's packed with these tiny membrane bubbles called synaptic vesicles.
Like little water balloons filled with something.
Exactly, filled with thousands of molecules of a chemical messenger, a neurotransmitter. The basic idea is the electrical signal arrives, triggers the release of this chemical, it floats across the gap, and then activates the receiving cell, the postsynaptic cell. And while we usually think of it as one-way communication, it's not always that simple. Sometimes the postsynaptic cell can send a chemical signal back across the synapse, things like nitric oxide in the brain. This is called retrograde signaling, and it can fine-tune the connection. It's more of a conversation, really.
A conversation using chemical words. Okay, let's break down that process. Boron and Boulpaep lay out 7 key steps for chemical transmission. Can we walk through those?
We sure can. Step 1 is packaging. The neurotransmitter molecules have to be loaded into those synaptic vesicles. This takes energy, often using a proton gradient, kind of like pumping them in.
Got it. Step 1, load the messengers. What's next?
Step 2, the arrival. An action potential, that electrical wave, travels down the axon and reaches the presynaptic nerve terminal.
Okay, the signal arrives at the doorstep. Step 3.
Step 3, calcium entry. The depolarization from the action potential opens voltage-gated calcium channels. These are specific doors that only open when the voltage changes. Calcium ions, Ca2+, then flood into the presynaptic terminal from outside the cell.
Calcium rushes in, and that's the trigger for step 4.
Precisely. Step 4, release. That surge of intracellular calcium is the critical signal. It causes the synaptic vesicles, which are docked and ready near the membrane, to fuse with the presynaptic membrane and release their contents, the neurotransmitter, into the synaptic cleft. They release these chemicals in distinct packets, or quanta.
So the messengers are released into the gap. Step 5 must be crossing that gap. Yep, step 5, diffusion. The neurotransmitter molecules simply diffuse across the short distance of the synaptic cleft.
And then they reach the other side, step 6.
Step 6, binding and activation. The neurotransmitter molecules bump into and bind to specific receptor proteins embedded in the postsynaptic membrane. Think of it like a key fitting into a lock. This binding activates the receptor.
Activating the receptor then triggers a response in the receiving cell.
Exactly. Step 7. Oh, wait. Step 6 is activating the postsynaptic cell via the receptor. And then step 7 is termination. You absolutely have to stop the signal quickly.
Right, you can't have the message just echoing endlessly. How does that happen?
3 main ways. Either an enzyme in the cleft destroys the neurotransmitter, like Pac-Man gobbling it up, or transporter proteins grab the neurotransmitter and pull it back into the presynaptic terminal or nearby cells for recycling. Or it simply diffuses away from the synapse. Which method is used depends on the specific synapse and neurotransmitter. Okay, 7 steps. Packaging, arrival, calcium entry, release, diffusion, binding, activation, and termination. Seems like a lot, but it happens incredibly fast.
Milliseconds in many cases. And the variety of neurotransmitters is huge small ones like acetylcholine, serotonin, glutamate, GABA, glycine, and larger peptide ones like endorphins. Each allows for different kinds of messages.
So when that neurotransmitter binds to the receptor on the other side, step 6, how does that actually translate into a response? How does the postsynaptic cell read the message?
Great question. It depends fundamentally on the type of receptor. There are 2 main functional classes. First, you have ionotropic receptors.
Ionotropic, meaning they directly affect ions.
Exactly. These receptors are ion channels. When the neurotransmitter binds, the receptor itself changes shape and directly opens a pore, allowing specific ions to flow through the membrane. This is super fast, we're talking milliseconds. It causes an immediate change in the postsynaptic cell's membrane potential.
Can you give an example?
The classic example is the nicotinic acetylcholine receptor at the neuromuscular junction where nerve meets muscle. Acetylcholine binds, the channel opens, lets sodium flow in and potassium flow out, causing a depolarization that excites the muscle cell. Many glutamate and serotonin receptors work this way too.
Okay, so ionotropic means fast and direct channel opening. What's the other type?
The other main type is metabotropic receptors. These work indirectly and are usually slower seconds to minutes. They aren't ion channels themselves. Instead, when the neurotransmitter binds, the receptor activates an intermediary molecule inside the cell, typically a G protein.
G protein.
Yeah, it's like a molecular switch. The activated G protein then goes on to trigger other intracellular events, maybe opening or closing separate ion channels, or activating enzymes that produce second messengers, which then have downstream effects. It's a biochemical cascade.
So indirect and potentially slower, but maybe more complex effects.
Exactly. And the beauty is illustrated perfectly by acetylcholine, or ISESH. We just said at the muscle, ISESH hits the ionotropic nicotinic receptor and causes excitation fast. But in the heart muscle, ASIC binds to a different receptor, a metabotropic muscarinic acetylcholine receptor. This receptor activates a G protein, which then specifically opens potassium channels, called GIRK channels. Potassium flows out, making the inside of the cell more negative hyperpolarization. This inhibits the heart cell, slowing the heart rate.
Wow. So the same neurotransmitter can excite one cell type and inhibit another, just depending on the receptor it binds to.
Precisely. It solved a big puzzle for early physiologists who saw ACES doing contradictory things. It's all about the receptor. Ionotropic receptors are often involved in fast excitation, like the nicotinic AChR letting GAtions in, or fast inhibition, like GABA or glycine receptors letting chloride ions in. Metabotropic receptors allow for slower, more modulatory effects.
Okay, this is fascinating. Let's really zoom in now on that prime example you mentioned, the neuromuscular junction, the NMJ. You said it's the most studied synapse. Why is that? What makes it such a good model?
Several reasons. It's relatively large and accessible compared to synapses buried deep in the brain. It's also highly reliable. Pretty much every time the nerve fires, the muscle contracts. And its basic mechanisms turned out to be foundational for understanding all chemical synapses.
So what does it look like? Describe the structure for us.
Okay, picture a motor neuron sitting in your spinal cord. It sends out a long axon, which then branches near the target muscle. Each branch ends on a single muscle fiber. The point of contact is called the endplate. The nerve ending isn't just flat. It forms these little swellings called boutons that sit in shallow grooves on the muscle fiber surface. And the muscle membrane underneath these boutons isn't flat either. It's dramatically folded into deep postjunctional folds.
Why the folds?
To massively increase the surface area. And packed onto the crests, the very peaks of these folds, is an incredibly high density of those nicotinic acetylcholine receptors. We're talking thousands per square micron.
Wow. So the receiving machinery is concentrated right where the signal arrived.
Exactly. The synaptic cleft, the gap, is about 50 nanometers wide here. It contains a meshwork of proteins, the basal lamina, and crucially, anchored in this basal lamina is the enzyme acetylcholinesterase, ACE, ready to break down ACE-alt immediately after it's released.
And the AC itself, where does it come from?
It's synthesized right there in the nerve terminal from choline and acetyl-CoA. Then it's actively pumped into those synaptic vesicles we talked about. Each vesicle is packed incredibly densely, maybe 6,000 to 10,000 AC molecules.
And where do these vesicles get released from? Is it random?
Not at all. Release occurs at specialized sites on the presynaptic membrane called active zones. These zones are rich in calcium channels and docking proteins, and they are precisely aligned directly opposite the mouths of those postjunctional folds on the muscle cell. It ensures the ACH is released exactly where the receptors are concentrated, maximum efficiency.
Incredible precision. So how did scientists actually figure out the electrical events happening there? How do you measure something so small and fast?
That goes back to the classic work of Fat and Katz in the 1950s. They used microelectrodes to record the voltage inside the muscle fiber right at the end plate. Now, normally the nerve signal causes such a large depolarization that it immediately triggers a muscle action potential, which masks the initial synaptic event.
So they needed a way to dampen the response.
Exactly. They used a low dose of curare, the poison arrow drug we mentioned, which blocks some, but not all, of the HEA receptors. This reduced the response size below the threshold for firing an action potential. What they then recorded was a transient, localized depolarization right at the end plate following nerve stimulation. They called this the endplate potential, EPP. It's a graded potential, meaning its size depends on how much HEA is released, and it fades as it spreads away from the end plate.
Okay, that's the voltage change. What about the current flowing?
For that, they used a technique called voltage clamp. This lets you hold the membrane potential at a fixed level and measure the current that flows across the membrane when the synapse is activated. This current is the end plate current, EPC. By clamping the voltage at different levels, they found something crucial. The direction of the current reversed at a membrane potential near 0 milliV. This is called the reversal potential.
What does that reversal potential tell us?
It tells you about the ions flowing through the activated channel. A reversal potential near 0 milliV means the channel isn't selective for just one ion, like sodium channels, which reverse near plus 55 milliV, or potassium channels, which reverse near and emit 90 milliV. Instead, the nicotinic ACE receptor channel is a non-selective tation channel. It lets both sodium ions, Na+, flow in, and potassium ions, K+, flow out pretty easily. There's also some calcium permeability. The net effect at the muscle's resting potential, around negative 90 milliV, is a strong inward current carried mainly by Na plus Eto, causing depolarization.
And why is being non-selective actually good here?
Because its job isn't to reach a specific ion's equilibrium potential, its job is simply to depolarize the muscle membrane above the threshold for firing a voltage-gated sodium channel-dependent action potential, which is usually around negative 50 milliV. Driving the potential towards 0 milliV achieves that very effectively.
That makes sense. Now, you mentioned earlier that neurotransmitters are released in packets or quanta. This sounds like a really fundamental idea. How was that discovered?
It's one of the cornerstones of neuroscience, pioneered again by Katz and his colleagues. While recording from resting muscle fibers without any nerve stimulation, they observed tiny, spontaneous depolarizations, much smaller than the EPP, maybe only 0.4 millivolts. They called these miniature endplate potentials, MEPPs, or minis.
It's happening randomly?
Seemingly randomly, yes. They showed these MEPPs were also blocked by curare and prolonged by drugs that inhibit ACE, confirming they were caused by EPA. The brilliant insight was that each MEPP represented the electrical response to the spontaneous release of ACE from a single synaptic vesicle.
One vesicle equals one MEPP.
That was the hypothesis. And they tested it beautifully. They knew that calcium entry was needed for the evoked release, the EPP. So they drastically lowered the calcium concentration outside the nerve terminal, or added magnesium, which competes with calcium. This dramatically reduced the amount of AC released by a nerve impulse. And when they did that, the EPP wasn't just smaller, it started to fluctuate in size in discrete steps. The smallest responses were the size of a single MEPP. Larger responses were clearly 2 times, 3 times, or 4 times the size of a MEPP.
Like counting packets.
Exactly. It was clear evidence that evoked neurotransmitter release is quantal. It occurs in discrete, multimolecular packets, each corresponding to the contents of one synaptic vesicle. This work earned Bernard Katz a share of the Nobel Prize in 1970. It fundamentally changed how we think about synaptic communication. It's digital, in a sense, built from these quantal units.
That's incredible. So synapses aren't just simple relay stations. Their strength can change. You mentioned modulation earlier. Facilitation, potentiation, depression. Can you briefly explain those again in the context of the NMJ?
Sure. These are forms of short-term plasticity, meaning temporary changes in synaptic strength, and the NMJ is a great place to study them. Facilitation is when you give 2 or more nerve stimuli close together in time. The second EPP is often slightly larger than the first. It's a short-lived effect, thought to be due to residual calcium from the first stimulus briefly boosting vesicle release for the second. Potentiation, or specifically post-tetanic potentiation, PTP, is a more dramatic and longer-lasting increase in EPP size. It happens after a period of intense, high-frequency stimulation, a tetanus. It can last for minutes and is linked to a more substantial buildup of calcium in the presynaptic terminal, making release more efficient for a while. And then there's synaptic depression. If you stimulate the nerve repeatedly at a high rate, the EPP amplitude can actually decrease temporarily. This is often attributed to the depletion of the readily releasable pool of synaptic vesicles, basically, using them up faster than they can be recycled and made ready.
So the synapse can get stronger or weaker depending on its recent activity history? That sounds like a basis for learning and memory in the brain, right?
Absolutely. These basic mechanisms of plasticity observed at the NMJ are thought to be fundamental building blocks for the much more complex forms of learning and memory that occur in the central nervous system.
Okay, we've talked a lot about these vesicles releasing their contents, being the basis for quanta. What's their life story? Where do they originate and what happens after release?
It's a whole cycle. The basic membrane components and some proteins are synthesized way back in the neuron's cell body, in the ER and Golgi apparatus. They bud off and are transported down the axon to the terminal. This is called fast axonal transport. Now, there's a difference depending on the neurotransmitter. Vesicles destined to carry peptide neurotransmitters are often filled during this transport process, but vesicles for small molecule transmitters like HA travel down empty. They only get filled with HA once they arrive at the nerve terminal, using specific transporters on the vesicle membrane.
So they fill up locally at the synapse.
Right. Once filled, they dock at those active zones, primed for release. When calcium enters, they fuse, release their AC, and then critically, the vesicle membrane doesn't just become part of the outer membrane forever. It gets retrieved via endocytosis, often involving a protein called clathrin that helps pinch off bits of membrane to form new coated vesicles. These recycled vesicles can then be refilled and reused. It's a continuous local recycling loop.
Very efficient. Are there key proteins that manage this whole docking, fusion, and recycling process?
Oh, absolutely. It's a molecular machine. We've already mentioned the calcium sensor, synaptotagmin. Then there are the snare proteins. These are essential for fusion. There's a snare on the vesicle, like synaptobrevin, a V snare, and snares on the target presynaptic membrane, like syntaxin and SNAP25, called T snares.
How do they work?
The snare hypothesis suggests they act like zippers or coils. The V snare on the vesicle interacts with the T snares on the membrane, forming a very stable complex that pulls the two membranes incredibly close together, overcoming the natural repulsion between lipid bilayers.
So they dock the vesicle and pull it tight.
Exactly, they prime the vesicle. Then, when calcium binds to synaptotagmin, it triggers the final step, possibly by interacting with the snare complex, causing the membranes to fuse and the fusion pore to open, releasing the transmitter. Afterwards, other proteins, including one called NSF, use energy to disassemble the stable snare complex, recycling the snares for another round. There are other important players, too, like proteins that link vesicles to the cytoskeleton synapsins and regulate trafficking, Rab proteins.
It's like a tiny, intricate factory running constantly.
Yeah.
Okay, we covered release. Now back to step 7, termination.
You mentioned acetylcholinesterase at the NMJ. How critical is that?
Absolutely critical for the NMJ. Acetylcholinesterase, ACE, is an incredibly efficient enzyme. It sits right there in the synaptic cleft, anchored to the basal lamina. As soon as AC binds to its receptor and comes off, or even just diffuses near AC, the enzyme hydrolyzes it, breaking it down into choline and acetate very, very rapidly.
Why so fast? To ensure the signal is brief and precise. If the AC lingered, the muscle would stay contracted or wouldn't be able to respond quickly to the next nerve signal. AC allows for rapid, high-frequency signaling needed for fine motor control. And cleverly, the choline that's produced is actively transported back into the nerve terminal to be reused for making more AC.
Recycling again. What about other neurotransmitters, like in the brain? Do they all have enzymes like AC?
Not usually. For most other small molecule neurotransmitters, like serotonin, dopamine, norepinephrine, glutamate, GABA, the primary termination mechanism is reuptake. Specific transporter proteins located on the presynaptic nerve terminal membrane or sometimes on surrounding glial cells actively pump the neurotransmitter out of the synaptic cleft and back into the cell. This clears the synapse and allows the transmitter to be repackaged into vesicles or metabolized intracellularly.
Okay, so either enzymatic breakdown or reuptake are the main ways to stop the signal. Now, given all this intricate machinery, it seems like these synapses would be vulnerable points, right?
Yeah.
Targets for drugs or toxins.
Highly vulnerable, and this has actually been incredibly useful for research and also clinically significant. Many toxins act very specifically on different parts of the synaptic transmission process. For instance, tetrodotoxin, TTX, from pufferfish blocks voltage-gated sodium channels, preventing the action potential from even reaching the terminal. Omega-conotoxins from cone snails specifically block the voltage-gated calcium channels needed for transmitter release.
And what about those famous bacterial toxins like tetanus and botulinum?
Ah, yes. Tetanus toxin and botulinum toxins, the source of Botox, are among the most potent toxins known. They are enzymes, specifically proteases, that get inside nerve terminals and cleave those essential snare proteins.
They cut the zippers.
Exactly. Different types of botulinum toxin cut different snares, SNAP25, syntaxin, or synaptobrevin. Tetanus toxin usually targets synaptobrevin, but specifically in inhibitory interneurons in the spinal cord. By cutting the snares, they block neurotransmitter release.
Which explains their effect.
Precisely. Botulism blocks AT release at the neuromuscular junction, causing flaccid paralysis muscles can't contract. Tetanus blocks the release of inhibitory transmitters in the spinal cord, leading to unopposed muscle contraction, rigidity, and spasms. But, as you know, the very specific action of botulinum toxin is now used therapeutically Botox to treat conditions with muscle overactivity, like spasms, dystonia, migraines, excessive sweating, and of course, for cosmetic wrinkle reduction.
Amazing how a deadly toxin becomes a treatment. What about drugs that target the postsynaptic side, the AT receptors themselves?
Lots of those too. We can have agonists, which mimic AT and activate the receptor. Nicotine is one, obviously. Acetylcholine is a clinically important one. It's used in surgery as a muscle relaxant. It activates the nicotinic receptors, causing an initial depolarization, but it's not broken down by AT, so it persists, keeping the membrane depolarized and inactivating voltage-gated sodium channels, leading to paralysis.
And antagonists, drugs that block the receptor.
Right. The classic is D-tubocurare, the main ingredient in curare. It's a competitive antagonist. It binds to the AC curare site, but doesn't activate the receptor, simply blocking AC from binding. This causes flaccid paralysis. Because it's competitive, its effects can be overcome if you increase AC levels, for instance, by using an ACE inhibitor. Pancuronium is a more modern synthetic version used in anesthesia. And then there's alpha-bungarotoxin from snake venom, which binds essentially irreversibly to the receptor, making it a powerful research tool.
So toxins and drugs highlight the importance of each step. What about diseases that affect this junction?
Sadly, yes. The most well-known is probably myasthenia gravis. This is an autoimmune disease. The patient's own immune system produces antibodies that attack and destroy nicotinic AC receptors at the muscle endplate.
So fewer receptors available to receive the signal.
Exactly. This leads to the characteristic symptoms: fluctuating muscle weakness and fatigability that worsens with activity and improves with rest. Often affects eye muscles, facial muscles, swallowing first. Because fewer receptors are functional, the MEPTs are smaller, and the EXP is less likely to reach threshold to trigger a muscle contraction, especially with repeated effort.
How is it treated?
Treatment often involves immunosuppressants to reduce the autoimmune attack, but a key strategy is using acetylcholinesterase inhibitors, like pyridostigmine. By slowing the breakdown of APO, these drugs increase the concentration and lifetime of APO in the synaptic cleft, giving it more chance to find and activate the remaining functional receptors.
Makes sense. Are there other NMJ diseases?
Yes. Another autoimmune one is Lambert-Eaton myasthenic syndrome, LEMS. Here, the antibodies attack the presynaptic voltage-gated calcium channels. So the problem is reduced ACU release rather than reception. It typically affects limb muscles more, and interestingly, muscle strength might actually improve briefly with repeated stimulation, unlike in myasthenia. And then there are congenital myasthenic syndromes. These aren't autoimmune, they're inherited genetic defects affecting various proteins at the NMJ. One fascinating example involves a mutation in the AC receptor itself in the channel pore region that causes the channel to stay open too long after AC binds.
Too much signal instead of too little.
Right. This leads to excessive depolarization, calcium overload in the muscle cell, and ultimately damages the endplate structure. It really highlights how the precise timing of channel opening and closing is critical for normal function.
Wow. So from the basic need to cross a gap to electrical versus chemical strategies, the intricate 7 steps, the specific machinery of the NMJ, the quantal nature of release, the receptor types, the termination mechanisms, and all the ways it can be affected by toxins, drugs, and disease. We've covered a huge amount of ground.
We really have. It's a journey through some truly fundamental physiology. Understanding these two types of synapses, the detailed steps of chemical transmission, the structure-function relationships at the NMJ, quantal release, ionotropic versus metabototropic receptors, and how signals are terminated, these are cornerstones. And as we saw, the clinical relevance is immediate. Neurological disorders, pharmacology, diagnostics. So much hinges on comprehending these synaptic mechanisms. It's not abstract theory, it's the operating system of your nervous system talking to your body.
Absolutely. And for anyone listening who's studying this, it might seem complex, but breaking it down step by step, like we try to do today, really shows that it's understandable. Each piece connects logically to the next. Physiology is this incredible interconnected puzzle. Keep digging into it, keep asking questions, and you'll find you absolutely can master these concepts. Thanks for diving deep with us today.